All right.
All right, so first up today we have Dr. Heather Walsh. She graduated with her PhD from West Virginia University in Forest Resource Science in December of 2018. Her dissertation focused on the use of molecular pathology techniques to understand mechanisms of disease in smallmouth bass. She's been involved in research at the USGS Leetown Science Center Fish Health Lab since 2006, where her main focus has been learning histopathology. So take it away, Heather.
All right. Can you hear me okay?
Sure can.
Okay. So I wanted to share this study I've been working on. This was part of my dissertation, and I saw a lot of the titles for this seminar series had to do with parasites and pathogens of fish, and I saw relevance in this study for the methodology that could be applied to some of the studies that you all have presented. And so let me downsize this. So my talk today is on the effects of tissue fixation on sequencing and transcript abundance of nucleic acids from microdissected liver samples of smallmouth bass. So to provide a little bit of background, we have a lot of, in our lab, we have a lot of formalin fixed and paraffin embedded tissues, FFPE tissues. And the benefits of having these is that they are archived samples of different types of nucleic acids from tumors, parasites, or different pathology associated with disease. But the drawback of having FFPE tissue or trying to use FFPE tissue in molecular applications is that fixation causes the nucleic acids to cross-link, specifically the formalin in the fixative. And then it's really difficult to use these samples in downstream molecular applications. And it's not always feasible to obtain frozen tissue, especially when you're field sampling, like 4 hours away from the lab, and it's hard to bring dry ice with you or liquid nitrogen. So although frozen tissue is the best for preserving nucleic acids, it's not always the easiest to do when you're out in the field. You can preserve tissue in ethanol or RNA later, which are great fixative preservatives for nucleic acids. But at the same time, there becomes a disconnect, and you cannot make a direct association between the pathology that you might be seeing on a regular histology slide or from the tissues that you preserved in the fixative and made that slide with, and then the nucleic acids that were preserved in RNA later or ethanol. So that's what this picture is kind of showing you here. You might have a chunk of liver and you weren't actually able to observe some type of pathology in the liver, whether it was a tumor or bile duct proliferation. But then once you get it back and you can look at it under the microscope, you're like, oh, look at that pathology associated with some kind of a disease. It'd be cool if we could do some gene expression studies. But sometimes that piece that you took in RNA later might not actually have that associated pathology in it. So you might have a disconnect there. That's where laser capture microdissection comes in. And the picture on the left is our laser microscope. And the picture on the right, on the bottom left, is how the laser microscope works. So the beam comes down through the objective, and you can actually cut out an area of tissue that's on the slide that you want to extract for DNA or RNA. You can cut out single cells, or you can cut out larger areas such as tumors or granulomas, preneoplastic areas such as bile duct proliferation or epithelial hyperplasia. And then you can also cut out a normal area so that you can compare the gene expression in both sections of tissue. You can also cut out parasites that you might not have observed macroscopically, such as myxozoan parasites that might be in the gills and you didn't actually see them when you were out in the field in order to take a sample of them. And then consequently, you can use these sections to extract for nucleic acids. So our laser microscope, you can actually place the microcentrifuge tube in a little rack that's outfitted on the microscope, and then you cut out your section and the tissue drops down into the cap of the microcentrifuge tube, and then you can extract DNA or RNA. So the objectives of this study specifically were to determine the effects of fixative type and fixation time on nucleic acid quantity and quality. We wanted to know, how does tissue fixed in formalin and non-formalin fixatives compare to controls? So the controls being 95% ethanol or RNA later. And how do these extracted nucleic acids perform in downstream molecular analyses? And I'm going to get more into these specific objectives in a little bit. So we sampled 5 adult smallmouth bass that had been reared in our laboratory, and we extracted the livers from each fish and took pieces of the liver and put them in 10% neutral buffered formalin, Z-fix, or PAX gene. So neutral buffered formalin and Z-fix both contain formalin, which can cross-link nucleic acids. However, PAX gene does not. It's a non-formalin fixative, more relatively newer, and it does not cross-link, so it's claimed to be better for preserving nucleic acids for downstream applications. And then, as I mentioned, the controls were RNA later and 95% ethanol. And these were fixed for 24 hours, 48 hours, 7 days, and 14 days. And the reason we chose some of these longer time periods, 7 and 14 days, is because sometimes when we're out in the field, if we're traveling to the Great Lakes, we're not getting back to the lab in 24 or 48 hours. So there's been instances where our samples have sat in the fixative for 7 to 14 days. And we wanted to know, are these archived samples that we have, are we able to use them for molecular analyses? So tissue sections were cut at 10 micrometers with the microtome. And as a lot of you know that work with histology, oftentimes sections are cut at between 4 and 6 micrometers. So this is relatively thick section. And the reason in doing so is that you can actually get more cells from a smaller piece of tissue. They were sectioned onto UV sterilized pen membrane slides, and these are slides that have a special membrane on the top that the tissue is dried onto, and then when you cut out the section, it drops down. It's still attached to the membrane, but the membrane does not interfere with any extraction methods. I did not stain the tissues. I did deparaffinize them prior to microdissecting them, and I have forgotten to deparaffinize them before, and it doesn't work very well with microdissection. Our microdissection laser scope is the LMD 6500, it's a Leica. And the size of the pieces were fairly large. I'd say they were maybe a little bit bigger than the size of a pinhead. So for nucleic acid purification, RNA was extracted from the LCM samples with the eZNA FFPE RNA kit, and the control samples were extracted with an eZNA total RNA kit. And all samples were quantified with the qubit RNA high sensitivity kit. For DNA, LCM samples were extracted with a proteinase K digestion buffer, and I took that from this methods paper by Lehman and Crape. It has tris HCl, EDTA, and tween 20 and proteinase K. And I did try out other kits with a spin column, but I was never, and I probably tried out maybe 4 or 5 other kits, but I was never able to obtain quantifiable DNA with those kits, only with this digestion buffer. And control samples were extracted with the QIAGEN DNeZ blood and tissue kit. All samples were quantified with the qubit double-stranded DNA high sensitivity kit. And DNA, or degradation of DNA and RNA, was assessed with the bioanalyzer. So to test the extracted nucleic acid, well, DNA in downstream application, I chose endpoint PCR, and I just looked at the EF1 alpha housekeeping gene. I developed this primer set that amplified a 395 base pair amplicon. For the LCM samples, I tried to use as much template as possible. So I used 10.5 microliters, which was the equivalent of 3 to 13.5 nanograms, which is fairly low, but it did work. For the controls, I was able to use a standard amount of template, only 1 microliter, which gave me 68 to 108 nanograms. And then I tested the samples with the Sanger sequencing on an ABI 3130 genetic analyzer, and analyzed the sequences with Genius. And I analyzed the quality of the sequences by assessing the Phred scores of a Q40 or higher. For RNA, I used the NanoString N counter, and this platform provides you with data, count data, that's very similar to what you would get with qPCR. And with this machine, you can actually multiplex and quantify up to 800 transcripts in a single reaction. For most of our work, our code sets are developed for 40 to 60 transcripts that we want to look at. And yeah, 800 transcripts is quite a bit, but I guess it is doable since they claim it can be done. Our smallmouth bass liver code set was developed in a publication by Hahn et al. 2016. It consists of 50 transcripts that are relevant to reproduction, immune function, contaminant metabolism, and it had a limit of detection of 16 transcripts. I only used 25 nanograms of purified RNA for every sample, including the controls. The recommended input amount is 50 to 100 nanograms that NanoString recommends. For all my other studies that I am able to use samples that had been preserved in RNA later, I do use 50 nanograms. So testing out this 25 nanograms was new to us, and it did actually work. So the statistics were done in R. I ran a Kruskal-Wallis one-way ANOVA with a Dunn's multiple comparison test and experience rank correlation analyses. These were to look at differences between the fixatives and how they correlated with the different attributes that were obtained. And then the transcripts were normalized in NSolver analysis software. The code set that comes with NanoString has some positive and negative controls in it, and then you have your housekeeping transcripts, so that's where normalization occurs. And I also looked at the housekeeping transcripts for their stability with a program called NormFinder. So here are the results. The mean RNA concentration of RNA later samples was about 800 times greater than samples fixed in the formalin and non-formalin fixatives. For neutral-buffered formalin and Z-fix, 24 and 48 hours were the best time frames, and as you can see, RNA concentration significantly dropped at 7 and 14 days, although I still was able to get some quantifiable RNA. And for PAXgene, the RNA concentration, I would say it was relatively stable over time. The highest time period obviously was 48 hours, but it didn't really seem like there was that drop off that we saw in the neutral buffered formalin and Z-fix samples. For DNA, mean concentration of samples preserved in ethanol, or the controls, was about 120 times greater. And compared to RNA, we got a lot lower concentrations of DNA. But that could be due to just the biology of a cell and the amount of DNA and RNA it regularly has in it. So let's see, Z-fix at 7 days actually had the highest amounts of DNA, and this went the same for neutral buffered formalin and PAXgene. This was somewhat of a surprising finding to me. I wasn't expecting this. I thought the results would be similar to what we got with the RNA, which would be 24 and 48 hours would give me the highest concentrations. And I also thought that samples preserved in patch gene would be higher than neutral buffered formalin and Z-fix. As far as the 7 days having the highest concentration, the only real speculation I can come up with for causing this is that Qubit says that as samples become more fragmented, their accuracy decreases. And at 7 days, the highest fragmented DNA was obtained. So that could possibly be a confounding factor here, but I'm not really 100% sure that that's what's going on. If anybody has any thoughts on that, I'd be happy to hear them. For RNA quality, this was done on the bioanalyzer by obtaining RIN values. RNA later RIN values were at least twice as high as the fixed samples for LCM. Now here, samples preserved in neutral buffered formalin had greater quality than samples preserved in Z-fix or patch gene. And Z-fix and patch gene were relatively similar as far as quality goes. So my guess as to why neutral buffered formalin samples at 24 hours had a lower quality could be that it wasn't a long enough fixation time, and maybe 48 hours to 14 days was better for the fixative to actually penetrate throughout the entire tissue. So there might not have been complete fixation of the tissues at 48 hours. For DNA quality, this was assessed by fragment size on the bioanalyzer. Interestingly, ethanol samples were not higher than fixed samples, and I'm not exactly sure why that occurred. Again, neutral buffered formalin samples outperformed the Z-fix and the patch gene samples for quality. However, as you can see, as I mentioned earlier, at 7 days, the smallest fragments were obtained. For Sanger sequencing, the mean high quality bases with a Phred score of 40 or higher, so this is the percentage. Ethanol samples were much greater. So in other words, their sequences were of much higher quality than the samples in the preservatives, and that is definitely to be expected. And here is where patch gene did kind of perform better than the formalin fixed samples. And between the neutral buffered formalin and Z-fix samples, those fixed for 48 hours had the best quality sequences. Whereas for patch gene, the best quality sequences were obtained at 14 days. So when the sequences were blasted in NCBI, a trend did show up with sequence length and the number of blast matches. So there was a significant positive correlation between the number of NCBI blast matches and sequence length. So the graph on the left shows that, and on the right, you see that NCBI blast matches, the percent is in the gray line, and then the black line is the sequence length. So at 14 days, when the sequence length significantly drops off for neutral buffered formalin and Z-fix, there were no blast matches to the EF1 alpha gene. Whereas when the sequence length is 500 or greater, there were 100% blast matches for the neutral buffered formalin at 24 hours, and a little bit lower of a percentage of blast matches as fixation, the amount of fixation, the time of fixation, I'm sorry, progressed. So the results for the nanostring encounter showed that the LCM samples, there was multiple transcripts with similar or greater transcript abundance to RNA later samples. And I was hoping that this would be the result, and I'm glad it was, because what it says is that although these samples were more degraded and fixed in formalin, they still worked as well on this platform as samples preserved in RNA later. And this is probably due to the fact that the company claims that FFPE samples work really well on their machines, because as I was talking about those code sets, the probes in those code sets that are for each probe represents each mRNA transcript that you want to analyze. And those probes are about 100 base pairs long. So as long as your fragments are 100 base pairs or greater, NanoString is going to be able to quantify them. So that is why it works well with FFPE tissue. And their claim to fame was true. For the LCM samples, so the PAX gene, there was one transcript that was significantly greater, significantly greater abundance than NutriBuffered formalin samples at 7 days. 5 transcripts with greater abundance than NutriBuffered formalin at 14 days, and 4 transcripts that were significantly greater than Z-Fix at 14 days. And I wanted to kind of emphasize this because, again, PAX gene says that the fixative is so much better at fixing samples for downstream applications. So it did see, there wasn't, in other words, there wasn't very much discrepancy between the transcript abundance of PAX gene samples and those samples fixed in formalin. There were actually differences in housekeeping and the other liver transcript abundances between the fixatives and fixation time. So what this means is that for each study that you or that we are going to pursue in the future, it would be in our best interest to optimize the samples and for their for which housekeeping transcripts work best with each type of tissue sample that is being used. So you wouldn't want to compare studies where some samples were fixed in like NutriBuffered formalin and some samples were fixed in Z-Fix, and maybe the ones fixed in NutriBuffered formalin were fixed for 24 hours and the one in Z-Fix for 48 hours. Because the differences that you might be observing between those samples might be just because the samples were preserved in different fixatives for different amounts of time, and not because of any treatment that was applied to those samples. The norm finder results showed that ribosomal protein L8 was the most stable housekeeping transcript. So the lower the stability rating, the better. Whereas the 40S ribosomal protein S12 was the least stable. So to summarize these findings, so what was the main points that came out of this study? The LCM samples had 800 times less RNA and 120 times less DNA than the controls preserved in RNA later and ethanol. For the nucleic acid concentrations, first place for DNA concentration goes to Z-Fix at 7 days. Second place for NutriBuffered formalin samples fixed for 24 hours. And third place for PAX gene at 7 days. So the two things that are not what I expected out of the outcome there was I thought PAX gene would probably be in first place and at 24 or 48 hours, not 7 days. And then for RNA, first place went to PAX gene this time for 48 hours, similar to what I would have expected. Second place was Z-Fix for 24 hours, and third place NutriBuffered formalin for 48 hours. So those results definitely made more sense to me. Nucleic acid quality, however, the best quality definitely went to NutriBuffered formalin for both DNA and RNA at 14 days. For DNA, second place went to PAX gene at 48 hours and third place to Z-Fix at 24 hours. And again, for RNA, second place went to Z-Fix at 14 days and third place went to PAX gene at 7 and 14 days. So the longer fixation times, I'm not really sure about why that's occurring. I would have thought it was 24 or 48 hours. But hey, the results are what they are. So to summarize this, PCR and Sanger sequencing were successful for these LCM samples, and sequence length influenced the number of NCBI blast match results, which was also linked to fixation time. So you would kind of expect, or you would definitely expect, that sequence length would have an influence on your NCBI blast match results. But it is interesting to find out, to know that it is also associated with fixation time. The nanostring encounter transcript abundance analysis was also successful, and it was successful with using only 25 nanograms of template. I've seen studies where they used a little over 6 nanograms of template, but as they pointed out in those studies, using less template may affect transcripts that are lowly expressed. So that should be definitely considered with what you're trying to analyze. If you know that your transcripts have high expression, there's probably not going to be any problem. But if your transcripts, the expression level is really low, you may need to try and obtain more template. RNA later samples had higher counts for many of the transcripts. There was variability in housekeeping transcripts, which, like, as I mentioned, will probably require optimization for different types of tissues and based on fixative and fixation time. And fixation time influenced the number of transcripts that fell below the limit of detection. Increase in number of transcripts below the limit of detection as fixation time increased, and PAX-gene showed more consistency in the number of transcripts below the limit of detection over time when compared to neutral buffered formalin and Z-fix. So just some final remarks of how these methods could be used in future studies and some of the things that we're planning on using it for. You can use it for endocrine disruption studies. As I mentioned, we use smallmouth bass quite often, so we're interested in trying to understand more about intersex in male fish. So we could do LCM of testicular cells, such as testicular oocytes, Leydig cells, or Sertoli cells. It would be kind of neat to know which cells are actually becoming testicular oocytes. We can cut out the gonad in young of the year fish. A lot of times these fish are so small and the gonad is so little, and it's hard to tell whether it's male or female. So with these applications, you could decipher that. And you can cut it out and try and use biomarkers to determine at the age at which, for example, smallmouth bass become induced with intersex. So there's applicability there too. You could cut out intrarenal cell hyperplasia from anterior kidney to try and identify stress markers. You can identify and explore transcripts associated with tumors. We have collected lots of white sucker and brown bullhead samples with different types of tumors associated with them. So we would like to go back and cut some of that out and analyze the gene expression in the tumors versus normal tissue. You can look at gene expression in neoplastic versus pre-neoplastic tumors, or you can even look at tumor progression and how these change. Sometimes, for example, we see skin papillomas, or we just see what look like large areas of epithelial hyperplasia. So it would be kind of interesting if we could get progression of these changes and analyze transcript abundance to see how they're associated and how gene expression changes. And we plan to use it for immunosuppression studies. We can cut out the anterior kidney in young of the year fish, which is very small and hard to isolate. So then we can go back, once we have a tissue section cut on a slide, we can go back and cut the anterior kidney out. Or we can look at lymphocyte proliferation, diffuse granulomatous inflammation, or focal granulomas like Mycobacterium. A lot of times granulomas have some valuable information in them, whether it's parasites or pathogens that they're trying to contain. And macroscopically, you might not see that these granulomas even exist. So if you can go back from your tissue section that you have for histology and go back and cut these areas out and then look at gene expression, you might get some answers as to what's causing these abnormalities. And then also, we're trying to use it for parasite identification. This area is kind of hard because a lot of times these parasites don't actually have a lot of DNA. So you have to, what we're trying to optimize here is how much tissue of these parasites we actually have to cut out. But it can be used for this. And there are studies out there that have done that. So the funding was provided internally by the USGS Environmental Health Mission Area. And with that, I will take any questions if we have time.
All right, thank you very much, Heather. Great presentation, very interesting. And I suspect there's going to be some questions, but we are over time already.
Okay.
So if, let me, all right, I just opened up the chat box. If you want to put your, if you're comfortable putting your email address in the chat box, if people have questions, they can just connect with you directly, if
Okay.
that's okay. So virtual round of applause for you. And if you could stop sharing your screen,
Okay.
I'll